In this episode of Toxicity Tests, we're going to look at genotoxicity. This first slide from the Alltox website gives us a definition of genotoxicity. It reads, germ cell mutagens genotoxins are substances that cause heritable, passed on to progeny, changes in the genetic material in germ cells, namely spermicides or oocytes. So here we're specifically looking at damage that chemicals or drugs can cause to the DNA in the sperm cell or the egg cell of females. It probably won't do you any harm just as you go about your everyday life. However, if you intend to reproduce, this damaged DNA could cause problems in your offspring. The rest of the paragraph reads, the term mutagen refers to a substance that induces transmissible changes in DNA structure involving a single gene or a group of genes. Genotoxins are a broader category of substances that induce changes to structure or number of genes via chemical interaction with DNA and or non-DNA targets. The term genotoxicity is generally used unless a specific assay for mutations is being discussed. This second slide is to do with regulatory requirements and test guidelines and reads, genotoxicity/mutagenicity testing is conducted for pharmaceuticals, industrial chemicals and consumer products, and the results are used to classify chemicals for heritable germ cell mutagenicity as well as carcinogenicity. Most regulatory agencies and international authorities recommend a test scheme consisting of in vitro and in vivo methods to identify genotoxic/mutagenetic substances. A tiered test scheme would likely start with computer-based prediction using quantitative structure-activity relationships (QSAR) and in vitro testing. The International Conference on Harmonization recommends a standard core battery for pharmaceuticals, which is undergoing revision at this time. The third paragraph reads, the working group nominated by the European Commission Services to review the status of genotoxicity testing for cosmetics concluded that the above in vivo methods, OECD TG 478, 483, 484 and 485, are not relevant for the purpose of the cosmetic industry. Apologies for this slide being a bit dry, but it's just passing on information. The final paragraph reads, the UN Globally Harmonized System classification of germ cell mutagenicity uses two categories. Category 1 chemicals are known to induce heritable mutations or to be regarded as if they induce heritable mutations in the germ cells of humans. Category 2 chemicals are those that cause concern for humans owing to the possibility that they may induce heritable mutations in the germ cells of humans. This slide describes the animal tests. I'm not going to read out the whole slide this time, but just to point out that there are several animal tests to test for genotoxicity. Under number 1 is OECD TG 478 and 485. And we've got TG483 and TG474 and TG486, and at the very last one TG484. So that's 1, 2, 3, 4, 5, 6, 7 or 8 tests to test for genotoxicity. The last sentence of this slide reads, in addition to these tests, other in vivo methods are being developed and validated for use in regulatory decisions, including the in vivo COMET assay and the in vivo transgenic mutation assays. This slide deals with non-animal alternative methods. It reads, a number of in vitro methods for genetic toxicity testing are based primarily on bacterial and mammalian cell assays, with several accepted by regulatory authorities. Testing proceeds by the use of a tiered test strategy using both in vitro and in vivo assays and providing information for predicting potential heritable germ cell damage as well as potential carcinogenicity. As well as there being a lot of in vivo tests, this paragraph, the second paragraph, says there's lots of in vitro tests to test for genotoxicity. It reads, 8 in vitro genotoxicity test methods have been adopted at the EU level with OECD guidelines, 4 of which are commonly used. These 4 in vitro assays include 2 mutagenicity test methods based on bacterial cells, the bacterial reverse mutation test, AIMS test, OECD-TG471, and the E. coli reverse mutation assay, OECD-TG472. The 2 commonly used methods based on mammalian cells are the in vitro mammalian chromosome aberration test, OECD-TG473, and the in vitro mammalian cell gene mutation test, OECD-TG476. Additionally, ECVAM validated the in vitro micronucleus test for genotoxicity testing in 2006 as an alternative to the in vitro chromosome aberration assay, revised OECD-TG487. Let's have a quick look at the Canadian Council on Animal Care's website to look at some of the alternatives in a little bit more detail. These first 3 slides I'll quickly flash up, but you can see that there are quite a lot of alternative tests, as the Alltox website has just said. However, let's have a look at 2 tests in just a little bit more detail. The first one is the in vitro micronucleus test, that's TG487, and the second one we'll look at is the in vitro COMET assay. This first test, I've just taken these 2 tests at random, it's called the in vitro micronucleus test, MNT for short. It says this is a genotoxicity test system used for the detection of micronuclei in the cytoplasm of interface cells to investigate the mutagenetic potential of a test compound in vitro. I'm not sure what micronuclei are, and I'm not sure what interface cells are, so I don't really understand the science of this particular test. However, under regulatory status it does say it's a draft TG487, so it must have some predictive qualities about it, and in the EU it says it's approved for the use as part of the REACH project. This second and last in vitro test that I'm going to look at is called the in vitro COMET assay. It says cells in culture are treated with the test compound. The cells are then lysed, which means they undergo something, and their DNA is run on a gel. If the DNA is damaged, the migrated pieces can be detected through staining. Under validation status it says Japan, JACVAM, which stands for the Japanese Center for the Validation Alternative Methods, presently is conducting a validation process. Under regulatory status it says that a new test guideline is proposed, and under effect or potential effect on animal use it says reduction, as it is part of a tiered testing strategy that results in the use of fewer animals. Before I go on to look at an essay on the Alltox website called Genotoxicity and Carcinogenicity, which alternative strategies, I'll look at one last slide from the Alltox website on validation. By means of shedding some more light on the validation process, I'm going to read out all 3 paragraphs on this slide. The first paragraph reads, in addition to the non-animal alternative methods already accepted and in use, ECVAM has coordinated the retrospective validation of the micronucleus test in vitro based on existing data. The ECVAM Scientific Advisory Committee endorsed the in vitro micronucleus test, MNT, which we've just been looking at on the Canadian Council on Animal Care's website, as a scientifically valid alternative to the in vitro chromosome aberration assay for genotoxicity testing following a weight of evidence retrospective validation. It goes on, the ESAC statement adds that the in vitro MNT can be considered for regulatory use as part of the Tier 1 genotoxicity test battery. The draft OECD TG487 for this method is being revised. An OECD ad hoc expert panel met in Atlanta, that was in October 2007, to finalise the guideline. The meeting was successful and the major issues of concerns were resolved. The second paragraph reads, the Interagency Coordinating Committee on the Validation Alternative Methods, that's the US Validation Center, has established the Genetic Toxicity Working Group, which is reviewing genetic toxicity test methods. The ICCVAM, could call it ICVAM for short, has not validated any new alternative methods for genotoxicity testing at this time. The last paragraph reads, an ECVAM panel estimated that the total replacement of animal testing for genotoxicity stroke mutagenicity at the EU level would take at least 12 years and require models for evaluating toxicokinetics and metabolism. We're now going to look at 4 slides from the Autox website from the essay Genotoxicity and Carcinogenicity, which alternative strategies? ? The subtitle is, why is it so hard to phase out in vivo genotoxicity? The first paragraph reads, genotoxicity is a crucial endpoint in safety assessment. In current guidelines, positive in vitro results are further investigated using in vivo assays, that's animal tests. Although numbers of in vitro assays have been around for decades, in vivo genotoxicity testing still has to be phased out. The main reason for that is the low specificity, many in vitro positives are irrelevant to human, of the in vitro genotoxicity assays. What the author is saying here is that the in vitro tests identify all the genotoxins, i.e. they have sensitivity, but they're not very specific in the fact that they also identify loads of drugs and chemicals that are not genotoxic to humans but do come across as positive in the genotoxicity in vitro assays. However, the same can be said of the animal tests as far as I know. I am basing my information on the carcinogenicity tests. However, I have no actual data on the genotoxicity tests, but an educated guess would be that they would be pretty much the same. So the state of affairs is that both the in vivo animal data and the in vitro data both have sensitivity but not specificity. You will notice that this essay was written in 2007, and since then the in vitro tests have come a long way. There's a company called Gentronix which has a new in vitro test which has both sensitivity and specificity, and I will look at that test at the end of this piece. For now though, I'll carry on looking at this essay because it does have some excellent information in it. Let's have a look at slide 2 now. The title of this slide is Combining in Silico and In Vitro Tools, a Step Towards Predictive Toxicology. Another way to tackle this issue would be to combine in silico and in vitro tools. It is worthwhile pointing to the fact that while performances of in vitro assays are usually checked, i.e. validated, those of in vivo, i.e. animal tests, are not readily available, i.e. they don't have to go through the same validation process, or they haven't been through the same validation process that the in vitro tests have. That's a very important point that she points out there. It goes on, similarly to the in vitro assays, the bioassays are providing information on hazard and not risk. To understand the difference between risk assessment and hazard identification, this slide may help a little bit. Imagine you're looking at arsenic. If you're looking at the hazard identification of arsenic, it's the determination of a substance's intrinsic toxicity, e.g. eye irritation, birth defects or cancer, through the use of toxicity tests. Basically what we're looking at through all of these toxicity tests. However, risk assessment, you also have to look at exposure assessment in the environment and the workplace, etc. So taking arsenic for example, you'd have your hazard identification data, but to work out risk you would also need to take into account whether there was any naturally occurring arsenic in the environment. Were people exposed to this arsenic, for how long, etc, etc. Back to this slide, it goes on, so why aren't in vitro results used for risk assessment? Well, I'm sure they are, but she says that they're not here. Anyway, we're getting a bit bogged down here. And it goes, exposure data, toxicokinetics and proper metabolic capability are lacking in simple cell systems. That sometimes is a fair point, if you see what I mean. Like if you're looking at toxicokinetics, That is about how a drug or a chemical enters the body and where it goes once it is in the body. And for that you need a systemic model, and this is why the animal is always used, even though it's not an accurate assimilation of the human model, the human being. But that's what she means there. Anyway, the author goes on, these are some of the types of information needed in addition to in vitro genotoxicity assays in order to perform risk assessment, i.e. she's saying the in vitro genotoxicity assays are not enough within themselves to perform accurate risk assessment. We'll have to look at that another time because it's such a big statement that is. A lot of the scientific anti-vivisectionists would say you're just getting misleading data from your in vivo tests. It's better to have a limited amount of accurate in vitro data and in silico data and data from epidemiology studies than it is to have confusing data from inaccurate animal models. But anyway, let's get on to the second paragraph. The first sentence of the second paragraph reads, most of the models, including the in vivo ones, i.e. the animal models, do not provide information about the sequence of events leading to the observed endpoints. They are descriptive. What she means here is, if an endpoint is cancer, we just described, yes, the mouse got cancer, or the in vitro tests has said you're going to get cancer from this because you've got many breakages, say, within the DNA. However, she's saying we do not know what the molecular, the tiny little points that led up to the cancer. We don't know that. Mechanistic is the word they use all through these things. We don't know the mechanistic steps. All we do is describe the end point. Oh yes, the rest of the paragraph pretty much confirms what I just said. What has been identified as key to using alternative methods is getting insight into mechanisms. And then it says, brackets, see report of the National Research Council, Toxicity in the 21st Century, a Vision and a Strategy. According to this report, risk assessment can become time and cost effective if we move from descriptive to predictive, i.e. mechanistically based toxicology. The third paragraph reads, huge amounts of data have been generated by several companies and agencies worldwide. Information systems have evolved and now are able to handle and process billions of data at once. The next move, which has already started, I'm going to have to correct the English on this sentence a little bit, would be to share data in order to make the best use of what has been done in the past and build the toxicology of the future. Data can be shared within consortia with a well-defined legal framework. Building quantitative structure-activity relationships, QSARs, is a way to secure the knowledge generated for decades with different in vivo and in vitro assays. I would warn against putting animal in vivo data into the QSAR models, otherwise you're going to get very confused. But human in vivo data, that is an absolute gold standard, and that should go into the QSAR models. This penultimate slide from the Alltox website and this essay reads, the Organisation for Economic Cooperation and Development has provided guidelines for developing transparent and mechanistically based models. So there is a consensus by all stakeholders, modelers, toxicologists, regulators and scientists in different fields on the properties of such models. In addition, the OECD is sponsoring a QSAR toolbox for registration, evaluation, authorization and restriction of chemicals, the REACH project. This toolbox will host not only QSAR models but also databases for read-across. This illustrates how important data sharing is in the field of alternative methods. The second paragraph reads, currently, in silico data cannot be used to stand alone. They are complemented by in vitro and or in vivo data. Again, I have to reiterate, it has to be human in vivo data. Anyway, it goes on. An example of the so-called integrating testing strategy combining in silico and in vitro approaches is illustrated by the works of Goebbels, Van Hal et al. The third paragraph reads, for in vitro testing, emerging technologies like toxicogenomics, high content screening and high throughput screening can help investigate the molecular mechanisms leading to specific endpoints. In the literature, several studies aiming at using toxicogenomics for predicting genotoxicity and carcinogenicity can be found. HCS-based in vitro micronucleus assays are available using automated image analysis platforms. Combining different in vitro approaches like toxicogenomics, HCS, HTS, and binding to nuclear receptors holds great promise in terms of unravelling molecular events. This is the goal of the US EPA, that's the Environmental Protection Agency's ToxCast program. Analysing the large amount of data that will be generated in this type of project is a real challenge. Here again, and here again I have to correct the English, here again a collaborative effort is needed in order to support such an initiative. This last slide gives the author's conclusions and perspectives. It reads, combining in silico with in vitro data may be a way to move towards animal-free genotoxicity and carcinogenicity testing. We are not there yet, but work is ongoing in the in silico as well as the in vitro areas. Initiatives like REACH and the 7th Amendment to the European Cosmetics Directive in the European Union, HPV High Production Volume Programme in the US and the DSL Domestic Substance List in Canada are incentives for seeking alternatives to animal testing. And the very last sentence reads, rather than having a defined framework, a flexible approach with an insight into the mechanistic pathways will be needed in order to generate relevant data. So now we're going to look at Gentronix, which is a company that has invented a new in vitro test to test for genotoxicity in drugs and chemicals. This slide reads, Green Screen HC is an accurate and fast in vitro human cell-based genotoxicity assay. The TK6 host cells are P53 competent and familiar to most genetic toxicology laboratories. A patented GFP reporter system exploits the proper regulation of the GAT45A gene. The second paragraph reads, the assay delivers both high specificity and high sensitivity and detects all common mechanistic classes of genotoxin. The 96 well microplate format takes about 20 minutes to prepare and delivers results after 48-hour incubation. A single microplate is used for the simultaneous testing of 4 compounds over 9 serial dilutions. The protocol is also readily automated using standard laboratory equipment. I'm now going to look at a peer-reviewed paper that was published in the journal Mutation Research / Genetic Toxicology and Environmental Mutagenesis, Volume 695, Issue 1-2, January 2010, pages 87 to 95. I'm just going to look at the abstract of this peer-reviewed paper on the Green Screen HC assay. The first half of the abstract reads, a recent ECVAN workshop considered how to reduce falsely predictive positive results when undertaking in vitro genotoxicity testing, and thus to avoid unnecessary follow-up with tests involving animals. As it was anticipated that modified versions of existing assays, as well as new assays, might contribute to a solution, an expert panel was asked to identify a list of chemicals that could be used in the evaluation of such assays. 3 categories of test chemicals were chosen, comprising a total of 62 compounds. This paper provides test results for these chemicals using the Green Screen HC assay. All tests were carried out in triplicate by multiple operators with and without S9 using invariant protocols. The results of the tests were as follows. Group 1 chemicals should have been detected as positive in an in vitro test. That means they should be detected as positive and they will or would cause genotoxicity in humans. And in the green screen HC test, 18 out of 20, that was 90%, were in fact identified as being genotoxic. In group 2, the chemicals should be negative. That means they should be identified as being not genotoxic. And in this particular part of the test, 22 out of 23 were in fact identified correctly as being negative non-genotoxins, and that was 96%. The overall concordance for groups 1 and 2 was 93%, which is incredibly high. Group 3 was a slightly different test. It says group 3 chemicals should give negative results in vitro mammalian cell genotoxicity tests, but have been reported to induce chromosomal aberrations or TK mutations in mouth lymphoma cells, often at high concentrations or at high levels of cytotoxicity. So again, really this is just another group 2 test, i.e. the chemicals should give negative results. And in fact 13 out of 17 did give negative results, so there was 4 false positives in this one, which was not quite as good, was it, as the group 2 test when there was only 1 false positive. However, that was 76%, which is pretty high still. And if we go to the last 4 lines, it says overall these predictive figures are similar to those from other studies with the green screen HC assay and confirm its high specificity, which in turn minimises the generation of falsely predictive positive results. To end this piece on genotoxicity, I'm just going to look at some of the press releases from the Gentronix website. This first slide says in July 2006, Gentronix secures an NC3R grant. It says Gentronix awarded a grant from the National Centre for the Replacement, Refinement and Reduction of Animals in Research. This is an institution that is financed by the government, so initially this company did receive some public funding. This slide talks about a press release in June 2009 about Gentronix and Apredisa, I think you pronounce that, allying to create a predictive toxicology service. I won't go into too much detail about it, but Dr. Katia Zion is the president of Apredisa, and she's also a scientific advisor to the Safer Medicines campaign. So I just thought I'd put that in there. Anyway, she's allied with Gentronix because she obviously, and she works in the private sector, she obviously has faith in this new green screen HC assay. And to finally end this piece on genotoxicity, we'll look at the last press release, which was from March 2010, and it says Gentronix signs a 3-year agreement with GlaxoSmithKline for green screen and blue screen assays. The second paragraph reads, Green screen HC was the first human cell-based assay technology that combined high sensitivity for detecting genotoxic chemicals with high specificity for reducing false positive results.